All right, let's get into it. Today, we're taking the journey deep inside our own cells to uncover the ultimate instruction manual for life. We're going to explore how a simple molecular code builds every single living thing you can see. It's an amazing story, so let's dive in.
So, have you ever really stopped to think about this? How does one single tiny cell contain all the plans to build something as complex as a human being, with a heart, a brain, and eyes? The answer is just, well, it's mind-blowing, and we're going to break down exactly how it works, step by step.
So the secret, it's all tucked away in a molecule called DNA. You should think of it as the master blueprint for an entire organism. It's locked up tight and safe in the cell's command center, the nucleus, and it has every single instruction needed to build and run the whole show.
Now, the cell is super smart about protecting this master plan. I mean, you wouldn't take the original priceless blueprints to a messy construction site, would you? Of course not. So the cell doesn't either. The DNA is the master blueprint. It's a stable, double-stranded helix. For everyday jobs, the cell makes a cheap, disposable copy called RNA. You can see the differences right here. It's single-stranded, uses a slightly different sugar, and it swaps out one of the letters, thymine for uracil. It's the perfect temporary work order.
Okay, so what happens when a cell needs to divide into two new cells? It has to pull off something absolutely incredible. It must duplicate that entire, massive blueprint flawlessly. This process is called replication, and getting it right is, well, it's a matter of life and death. Let's see how it's done.
The whole thing really boils down to three main jobs. First, you have to unwind. The two intertwined strands of DNA have to get unzipped. Second, you copy. A new, matching strand is built for each of the two original strands. And finally, the most important step, proofread. The cell does a quality check to catch any mistakes.
First up on the team is an enzyme called helicase. We can just call it the unzipper. It has one job, but it is so important. It moves down the DNA molecule and just unzips it, breaking the bonds holding the two strands together so the code can be read.
Next up, we have the star of the show, DNA polymerase. This is the master builder. It zips along each of the original strands and adds the correct matching letters, or bases, building a brand new strand. And this thing isn't just fast, it's a perfectionist. It even has a built-in proofreading function to check its own work as it goes. Incredible, right? Now, here's a little wrinkle. DNA strands have direction, kind of like a one-way street. This means our master builder, DNA polymerase, can build one of the new strands in one smooth, continuous piece. But the other strand is like a one-way street going the wrong way. So polymerase has to build it backwards, in these little chunks called Okazaki fragments. And that's where our next player, DNA ligase, comes in. It's the gluer. It comes in and seals all the little gaps between those fragments, creating one perfect new strand.
All right, so that's how you copy the entire library for a new cell. But what about for just day-to-day jobs, like making one specific protein? You don't need the whole blueprint for that. You just need a single page. This process of copying just one gene is called transcription.
This is exactly what transcription is. It's the process of making that working copy we talked about earlier. It takes the instructions from a single gene on the DNA and writes them into a portable little molecule called messenger RNA or mRNA. This mRNA is the work order, ready to be sent out to the factory floor of the cell.
And the molecule that does this job is called RNA polymerase. You can think of it as the cell's scribe. It finds the start of a gene on the DNA, reads the code, and writes down a matching mRNA message. It's basically translating from the language of DNA to the language of RNA.
Now, in complex cells, like ours, the first mRNA message that gets made is kind of like a rough draft. So before it leaves the safety of the nucleus, it gets a little editing. A protective cap is put on the front, some junk sections called introns are cut out, and a long tail is added to the back for stability. After all that, the message is finally ready for prime time.
Okay, our little mRNA work order is all polished up and ready to go. It leaves the nucleus and heads out to the cell's protein-building factories. And this final stage is called translation. This is where we go from the language of genes to the language of proteins, the actual building blocks of life.
The language of mRNA is read in three-letter words called codons. It's basically a dictionary. Each three-letter word stands for one specific amino acid, which are the building blocks of proteins. So when the factory sees the codon AUG on the mRNA, that's a crystal clear instruction, bring me the amino acid methionine.
So you've got this four-letter alphabet, A, U, G, and C, and you're making three-letter words.
That gives you 64 possible combinations, or codons. And here's what's so cool about this. There are only 20 amino acids. This means some amino acids have more than one codon. It's a built-in safety net. A small typo in the DNA doesn't always change the protein, which makes the whole system much more robust. Oh, and 3 of those codons are just stop signs, signaling the job is done.
And where does all this happen? In a molecular factory called the ribosome. This incredible little machine clamps right onto the mRNA message and starts reading it codon by codon to build the protein chain exactly as instructed.
Translation also has 3 basic steps. First, initiation. The ribosome finds the start signal. Then, elongation. The ribosome chugs along the mRNA, and as it reads each codon, a little delivery molecule called transfer RNA, or tRNA, shows up with the exact right amino acid. The ribosome links them all together in a chain. And finally, termination. The ribosome hits a stop codon and, poof, it releases the brand new finished protein.
So there you have it, the central dogma of biology. DNA makes RNA, and RNA makes protein. But why does all this matter? I mean, really, why should you care about this intricate process? Because it is the foundation of our health, our diseases, and some of our most powerful new technologies.
Well, because when this beautiful, elegant system breaks down, the results can be devastating. If the proofreading system fails during replication, it can lead to mutations that cause cancer. A tiny mistake in splicing that mRNA can cause genetic disorders like beta thalassemia. The accuracy of this process is, quite literally, a matter of life and death.
But here's the flip side, and this is where it gets really exciting. By understanding the blueprint, we can actually hack the code for good. We can design antibiotics that specifically jam up bacterial ribosomes, killing infections without hurting us. We can copy DNA in a test tube using PCR to diagnose diseases. We're even creating medicines that deliver custom mRNA instructions to ourselves. This knowledge is the bedrock of modern medicine.
And get this, here's a little mind-blowing fact for you. For decades, scientists figured proteins did all the real work in the cell. But it turns out that the actual engine of the ribosome, the part that forges the bonds between amino acids, isn't a protein at all. It's made of RNA. It's something called a ribozyme. Just goes to show how incredibly versatile this little molecule really is.
So yeah, from a simple 4-letter code to the incredible complexity of life, this entire process is just so elegant and precise. And we've gone from just observing it to being able to read it, and now even to writing and editing it. And that leaves us with a huge question for our future. Now that we finally understand the blueprint of life, what should we build next?